Competition is at the heart of all life forms. In reproduction, however, the competitors are less than 1/100th of a millimeter wide. In vitro fertilization, or IVF, made parenthood possible for couples struggling to conceive by fertilizing a woman's egg in a dish and placing it back in the womb. However, sperm quality continuously deteriorates over the last decades, and many sperm cells, despite being motile and capable of fertilizing an egg, would not produce a healthy embryo. Therefore, embryologists often select a sperm cell by themselves and directly inject it into the egg. This process is a special type of IVF called intracytoplasmic sperm injection, or ICSI. But this procedure also lacks the natural mechanisms in the woman's body. How would the clinician know which is an adequate sperm that should fertilize the egg? The problem is that sperm cells are nearly transparent, therefore almost invisible under regular microscopy. Apparently, there are established criteria for high-quality sperm morphology only for stained cells. However, staining the cells with visible dye is not allowed, as it might damage the cell. Today, the sperm selection process relies on poor image quality, and therefore the results are highly subjective, and only very conspicuous abnormalities are detectable. There is also no ability to evaluate the DNA integrity of the selected cell. During the last years, Professor Natan Shaked and his research team at Tel Aviv University developed a novel optical imaging system that can visualize these transparent cells in real time, 3D, and without staining. As opposed to regular microscopy, this system records how much light is delayed when passing through the cell. This tiny light delay is impossible to measure with a regular microscope. But if we record the addition of a low-power laser light beam passing through the sample and a clear reference beam that does not contain sample details, a digital hologram is acquired and processed to the optical thickness map of the sample. This quantitative map can be used to accurately analyze the sperm in 2 and in 3 dimensions, without staining and without damaging it. The research team has published that this new stain-free technique can be performed at the same level of accuracy as stain-based analysis. Furthermore, the new technique is quantitative and therefore objective, as it allows obtaining a novel set of cellular parameters previously unattainable to the clinicians, such as cellular mass and volume. These new parameters would provide critical information about the cell, such as its DNA integrity. Until now, holographic imaging capabilities required expensive, complicated equipment, and therefore were out of reach for IVF clinics. But the research team ingeniously managed to downsize the system to a small, simple, and elegant box, to be operated directly by the clinician with manual or automatic single sperm cell selection. Based on these new capabilities, QART was founded. We aim at commercializing this technology and obtain automatic analysis, sorting, and selection of sperm cells for fertility clinics worldwide.